Hello everyone. So my name's Ed, and I work in FutureNeuro over in our CSI at Stevens Green. So today, what we're going to do is go through a recent project that we published that was studying the role of the gut microbiome in a rare form or a rare condition that leads to epilepsy, known as limbic encephalitis. Hey, right, okay. So very quick background. That's the picture of the paper. It's the title of the paper, as well as where it was published in a journal called Epilepsy. That's my name, I guess. So, limbic encephalitis. So this is a rare disease that leads to epilepsy. It is essentially the immune system mistakenly attacking parts of the brain. So, as the immune system interferes with brain activity, this can lead to epilepsy. And what we've got is proteins that are binding to receptors in the brain cells. So brain cells communicate with each other by either sending kind of packets or bubbles of chemicals to each other, or by physically interacting, kind of like a key and lock mechanism of proteins binding to each other. If we interfere with that mechanism, we can lead to neurological disease, which is what is happening in limbic encephalitis. There are two predominant forms. And they're based on the receptor protein on the brain cells that are being attacked by the immune system. So there's LGI1, limbic encephalitis, and there's Caspr2. They have slightly different presentations because the immune system is attacking two slightly different proteins that may have slightly different functions in the brain. We're going to focus on LGI1 for the purpose of this presentation. It's the predominant form, and the majority of the individuals that we studied as part of this study were LGI1 positive. So that's the brain cells. The immune system. So there are a cluster of genes on chromosome 6 that are known as the HLA genes. It stands for the human leukocyte antigen gene locus. And there's a cluster of lots of different genes that together form these receptors on all the cells in your body that recognize non-self sources of protein. So basically stuff like bacterial or viral infections. So when you have something that shouldn't be in your body, parts of that cell or that organism are going to be broken up, and your cell will then take these fragments, kind of like discarded skin cells for us, and present them on a cell. And the immune system comes towards them and notes that you've got something that's outside of the body that has managed to get in from this sort of signaling, and then attack what should be something that shouldn't be there. What is happening here is that our HLA genes are slightly modified by genetic mutations, and so they are erroneously presenting things that are actually some materials from your own body. So basically, this is how the immune system accidentally starts attacking our cells. So this happens in a lot of autoimmune diseases. So, for example, forms of arthritis, as well as lots of other autoimmune disorders. So these are essentially presenting what are known as antigens, these materials that provoke an immune response. And they should be, usually, something that's not from ourselves. So HLA genes have lots of different subtypes, basically by different mutations that are in these genes. There are lots of genes that form the HLA gene cluster. One of them is called HLA-DRB1, and it has a particular subtype that's known as 0701. This is just a nomenclature system that we've come up when we detect them. We and other people have found that this particular subtype of DRB1, 0701, is really common in individuals with LGI1, limbic encephalitis. So this is data from our collaborators as part of the paper based in Oxford, Drs. Irani and Binks. And they found that there's, what I've highlighted in these red boxes, along the red bars are different forms of HLA genes and their frequency in individuals with either LGI1 positive or Caspr2 positive limbic encephalitis. And then in black bars are our healthy controls, individuals that don't have this disease. And you can see that there is a particular bar that's very common in our LGI1 positive. So this corresponds to that HLA-DRB1 0701. But interestingly, it's really common in our healthy controls. So if this form of this HLA gene was the sole cause of this really rare form of neurological autoimmune disease, it doesn't really make sense that it's quite common in the general population because they don't have this really rare form of disease. So essentially what we're doing is trying to search for this missing puzzle piece. So one, it's found in 90% of LGI1 patients, so it could be a plausible cause, but healthy individuals carry it in high frequencies as well. So it can't be causal on its own. So we're trying to find basically this sort of this missing trigger that bridges the gap between this genetic risk that leads to this rare form of disease. A potential answer is the gut microbiome. So this is a term for all of the bacteria, viruses, fungi, and a special type of bugs called archaea that live in the gut, in the digestive system. These are all microorganisms. Everyone has it. It's very useful and very useful for life. So there's a lot of healthy functions that the gut microbiome carries out. So for example, when you eat things, your own genes, your own proteins are helping digest that food. But there are a lot of that digestion is done by your gut microbiome as well. So it's really useful for healthy digestion of food. So this plays a normal role in nutrition and in health. And it interacts with the host body, i.e. ourselves. But there can be dysregulation or disbalancing of the composition of the gut microbiome. And when it gets dysregulated, it can lead to disease. So an unbalanced gut microbiome could be either a loss or deficit of beneficial bacteria, or it could be an overgrowth of potentially pathogenic or disease-causing bacteria. You can also have what's known as a loss of general bacterial diversity, which is also bad in itself. A healthy gut microbiome is a diverse gut microbiome. It's recognized to raise the risk of inflammatory bowel disease, for example. This is one of the diseases, one of the inflammatory diseases, rather, that has a known component where the gut microbiome and dysregulation in the gut microbiome leads to the disease. The idea being that perhaps the gut microbiome plays a role in other inflammatory diseases, including neuroinflammatory diseases like limbic encephalitis. So this is where Epilepsy Ireland comes in. So you guys funded this project, which we are very grateful for. This helped me as a postdoctoral researcher. I remember starting this project down in lockdown in the basement flat down in Rathmines, and life has moved on since. And we can now present the results of the recruitment that we were carrying out then. So let's talk about study design. What we are doing essentially is testing the hypothesis that the gut microbiome is this missing piece of the puzzle, this kind of triggering of individuals that already have a predisposition thanks to a genetic variant in the HLA genes. So what we did was recruit 42 patients with LGI positive limbic encephalitis. We also recruited individuals with Caspr2 positive, but there were only about 4 individuals that we were recruited. This is a very rare disorder, so we're talking about quite small sample sizes. We also matched them with 24 healthy controls, and these were family members. So the idea being they share the same environment, same dietary and same kind of living environment. They should have a similar gut microbiome. So any differences in the gut microbiome might be due to the might be a triggering change in the gut microbiome that could lead then to the disease. So we collected 2 DNA samples. We collected genomic DNA from the individual, i.e. their own personal genome, to study, for example, if they had the genetic variant, this HLA variant. And then we took stool samples and extracted the DNA from stool samples. And this would provide us with the DNA of their gut microbiome. So what we did, well, for each of these stool samples, we extract the DNA from that stool sample, and then we sequence that DNA. And we basically have a load of fragments of DNA that we've collected from the gut microbiome. We can do 2 separate things with this DNA. One, we can investigate the species that we observe in that sample. So we can reconstruct the genomes of the species, of all the bacteria, viruses, and so on, within that stool sample. But we can also instead look at the actual genes that are present within the stool sample. So bacteria do something very interesting, and that they share their genes quite a lot in something known as horizontal gene transfer. So unlike us, where we have a load of chromosomes that all contain our genes, bacteria sometimes have a lot of small circular genes, small circular fragments of genomes that contain a small number of genes, and they can swap these with other species, completely different bugs, and gain completely different new functionality based on them. So sometimes it's useful to just look at the functionality that is in the gut microbiome as well. Bacteria are very complicated. I like studying humans usually. So first of all, we looked at compositional changes. So this is not looking at the function, this is just looking at the known species that are present in the gut microbiome of these individuals. So how on earth do we test these differences? So this is just a schematic of all of our individuals along the x-axis. It's broken up by either whether they are an LGI positive case or whether they are healthy control. And then we've got a stacked bar plot. So each one of these stacked lines is representing one individual, and the proportion of colours represents the proportion of species that we observed in their gut microbiome linked to the types of species. And you can see that there's a massive diversity, but visually there's not much of a clear difference between the two of them. So how can we test these statistically? What we can do is look for diversity. So we can measure the diversity of species within a sample. And we find that in one particular measurement of diversity, the people with LGI1 positive, or who were LGI1 positive, had a slight reduction in bacterial diversity compared to their healthy controlled partners, which may suggest that they might have a slightly more unhealthy gut microbiome, perhaps. We can also look at inter-sample differences, so we can see whether, in general, our cases and controls look different as two separate groups. But there wasn't very much clear distinction between cases and controls. So, for example, if we just characterize people by the types of species that we see in them, we didn't see this really clear distinction between our cases and controls. I.e., there's no large-scale differences observed. So if we sequence individuals who have this condition, and we sequence their gut microbiome, there's not going to be this gut microbiome-wide signal to predict whether they're going to have this condition or not. So whatever is going on in the gut microbiome, if there is something, is going to be far more subtle. So what we can do is look for individual species. Maybe there's a particular species that are overrepresented in our cases versus controls, and perhaps then that species is a plausible trigger. We didn't find any individual species that were significantly associated with our cases and controls. This is probably down to really small sample sizes. We're sequencing and comparing 42 cases versus 27 controls, and this is kind of hampered by the fact that it's a rare disease, and to take part in the study, you do have to provide a stool sample. So there are a couple of barriers to gaining sampling. So we find that there are some species that have maybe tentative evidence of being enriched in our cases or depleted in our cases, but no real smoking gun within individual species. We can also look at just general compositional changes rather than just looking at total diversity or individual species. We can look somewhere in between. We found this interesting signal where we can class the majority of bacterial species in the gut microbiome as belonging to two branches of the evolutionary tree. One of them is the Firmicutes, and one of them is the Bacteroidetes. I've had a lot of practice saying these species, or these taxa names. We found that the Firmicutes, or species that belong to this Firmicutes branch, are enriched in our controls, or they're depleted in our cases, depending on which way you want to think about it. And the Bacteroidetes were enriched in our cases, or depleted in controls. And it looks like there's some sort of change in the balance of these two different phyla. Now, these are two really big groups of bacterial species. We're not talking about individual species of a particular genetic background. We're talking about large-scale, kind of subtle differences, but something that looked interesting. So we looked into it a little bit more. So we do find that there's this balance of Firmicutes and Bacteroidetes is lower. So we can measure this as an FB ratio, essentially the ratio of species belonging to Firmicutes and belonging to Bacteroidetes. And we find that the FB ratio is lower in individuals with LGI positive limbic encephalitis compared to their healthy control family members. What was very interesting is that we then looked at that HLA genotype for these individuals, because this is one of the, if not the genetic profiles that is predictive in someone that has this. We found something interesting. In our healthy controls on the right, there is no relationship between the FB ratio and their genotype. So whether how many copies of this HLA gene, that they altered HLA gene that they carry. But within our cases, we see this interesting relationship as the more copies of this mutant form of HLA they have, the lower the FB ratio is. Basically, the more that they have this genetic risk, the lower this ratio, this signal is, which is quite interesting. We're going to need to really replicate this in other studies, but it's one of the first sort of like compositional changes that we're seeing in this disease. So that's compositional changes. What we also looked at was functional changes. So as I mentioned, what we can do is characterize someone's gut microbiome by the species that belong in it or that habitually live there, but we can also characterize the gut microbiome in terms of the functions that those those bacteria carry. So instead of placing the DNA that we detect in terms of species, we can place that DNA in terms of genes that are present. So genes are our functional units within genetics. Bacteria mix and share genes quite happily. So we don't find evidence of a specific pathway that's enriched in our cases and controls. Again, really small sample sizes. So it would have to be a really strong signal of a particular species or a particular genetic pathway that's being enriched in our cases. And we don't see evidence of a really significant shift within the gut microbiome of these individuals. But we did find a really common theme between metabolism of different chemicals in the gut microbiome that lead to the production of what's known as short-chain fatty acids, or SCFAs. These are really interesting within a neuroinflammatory context. So these are small molecules that are derived from the metabolism that these microbiota carry out. So these are small molecules that are generated essentially by the gut microbiome happily munching down on all the food that you've sent it into your digestive system. These short-chain fatty acids can then be taken up by the body through the gut lining and then used, in your own biology. And they appear to have an anti-inflammatory effect. In lots of different inflammatory diseases, it's known that short-chain fatty acids are associated with this kind of protective anti-inflammatory behavior. And there's also some evidence that short-chain fatty acids provide some protection against seizures and maybe anti-neuroinflammatory as well. So we know that short-chain fatty acids have evidence of anti-inflammatory behavior, but they also specifically have some evidence being neuro-anti-inflammatory. So, short-chain fatty acids appear to be reduced in our LGI1 positive cases, although individually for those pathways we don't have statistical evidence. So we're going to have a larger sample size, perhaps have a separate study in a different sample of LGI1 positive and then combine them together and see whether the signal remains. But it's really, really interesting and really encouraging that the sort of signals that we're seeing as part of these functional changes are to do with inflammatory behavior. So hopefully with larger sample sizes, we're going to be able to start teasing out the role of this gut microbiome, because there appears to be something in it. So, concluding remarks. We didn't find any individual bacterial species enriched or depleted in our cases and controls. We didn't have a lot of sample sizes just because it's a rare condition. We do find, though, that people with LGI1 positive limbic encephalitis have fewer Firmicutes species in general than they have compared to their healthy control partners. And this shift is far more pronounced when you take into account their individual's genetic risk loci. So this already appreciated genetic risk factor that raises your risk of limbic encephalitis. And that genetic risk factor is really common in the general population. So it appears to be that there might be indeed a role that the gut microbiome is essentially adding a second hit to that genetic background, leading to risk for this trait. People with LGO1 positive limbic encephalitis also appear to have a reduction in short-chain fatty acid production in their gut microbiome. So it appears that the bugs that they do have in their gut microbiome don't produce as much short-chain fatty acids as their healthy control partners. And short-chain fatty acids are neuroprotective, or at least anti-inflammatory. So this seems to be consistent with this idea that there is some sort of innate inflammatory background in the gut microbiome that may be leading to this disease. But I do need to stress that we don't have enough sample sizes to characterize the smoking gun. So we're going to need more samples. This is a rare disorder, so we have near complete ascertainment within Britain and Ireland for this rare disorder, working with our colleagues in Oxford. So we're going to need to collaborate with international collaborators and start building together joint data sets. The challenge for gut microbiome or microbiome studies is that there is a large impact of batch effects between sequencing stool samples between different individuals. So a study procedure might immediately sample gut microbiome within, say, for example, a clinic, and those samples immediately go to a biobank for DNA extraction and processing. Or we might be doing it via remote postage, i.e. someone's doing it in the comfort of their own home and then posting those biological samples to us, which might take a day or two. And these changes of time between sampling and DNA extraction have large effects on gut microbiome sequencing results, just because the biological samples are less stable and so on. So this basically means that combining data sets with the gut microbiome is still quite difficult, and is still sometimes the sampling scheme for these studies means that restricts the amount of data sets that we can add together and jointly analyze to improve. So this is one of the challenges within the gut microbiome of studying it, but as more studies are released and we kind of get a more kind of consistent sampling scheme and able to control for what's known as these batch effects, we should be able to increase sample size nicely and start seeing whether these kind of anti-inflammatory signals or loss of anti-inflammatory signals in the gut microbiome of these individuals is a true signal. Thank you very much for listening. I'd like to thank all the LE patients and the families for taking part in this study. As it's a gut microbiome, it's asking a lot from them. Obviously like to thank Epilepsy Ireland for funding this for the study, everyone in FutureNeuro that contributed to the study, and then everyone else that helped out with the project and the analysis.